Welcome to the SBGrid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SBGrid.
Once again, welcome to the last SBGrid webinar of this academic season. We have Mart last year to talk about segmentation with sum topogram averaging, modeling cellular architecture. If you have questions, feel free to send them to the hosts in chat. Use the Q&A button. Raise your hand. We will go from there. And with that, I will turn it over to Mart. You're not here to hear me talk. So.
All right, thanks, Pete. Well, hello, everyone. Hello, list of names. Next slide. I thought I'd introduce myself first and my background to explain where I'm coming from. So my name is Mart Solast. When Pete et al. Invited me, my name was Mart Last, so non-taken Pete, but it's been updated. And I'm currently a postdoctoral fellow at the MRC LMB in Cambridge. My background is in applied physics. I studied a thing called nanobiology and applied physics later on in Delft in the Netherlands. I worked for Belmont for a little while on correlative microscopy. Did my Ph.D. in Leiden, working on more correlative microscopy, and I'm now a postdoc with Matteo Allegretti. And my interests are two things. The first half being method and software development for cryo-electron tomography. So this video over here is one of ICE, which is a toolbox for segmentation, training neural networks, segmenting and particle picking. And then I try to combine this software development with using that software and other tools to do some novel biology myself. So this is an example of my postdoc project, which is about metabolic channeling and purine synthesis. And specifically this protein over here, which is INPDH, which I'll get back to later. So that said, I will talk mainly about segmentation. And I've noticed that often when you mention segmentation, people's first thought of the use of segmentation seems to be that it's for visualization, which is true and definitely very useful, but there is more to it. And in my workflow that I've sort of landed on after a couple of years of playing around with segmentation and then focusing on subtomogram averaging for the last year or so, is that you can use segmentation for a lot of stuff. So one of those, or one of the applications is particle picking, where you segment and then you turn your segmented mesh or volume into coordinates. You can filter particles. So if you know that a particle occurs specifically in mitochondria, now if you can pick your particle with sample meshing, something like that, and you can segment mitochondria, and this is done by people, they often do it manually. But if you use automatic segmentation for that, you can constrain your particles to specific environments. There's visualization. There is priors for identification. So if you can detect multiple features and you already know biologically that particle A always sits on top of particle B, then the detection of these two different things helps you identify. Priors for orientation. Example here is a nuclear pore complex, where if you have a coordinate for a nuclear pore complex, but you don't know which way it's facing, if you could simply detect nucleus versus cytoplasm, that can give you information on how to orient the particle. And then finally, getting around large data sets with a tool called POM. And I'll talk about ICE, the segmentation suite, first, POM second, and then later on about easymode, which is a little more recent. So, POM. Like I said, ICE is a tool to train your own networks. And the idea there is to streamline the whole process and make it a little bit easier to land on weights for a neural network that work for your data, and then to use segmentation in your workflow for your data specifically. POM is visualization, contextualization, and curation, and easymode pre-trained networks. If you're interested in reading about any of these, we have a paper and 2 preprints online, which I'll list again later. So starting with ICE, I thought I would just run you through it by showing you the whole workflow in a couple of short videos. So essentially, if you want to pick something and you want to do it via segmentation, which I would recommend because it can be a pretty interactive and fun way to engage with the data and sort of explore what's in there. If you want to use that sort of approach for your own data, what you'll need is a neural network that gives you decent output on your data. The way to do that in ICE is you would open ICE, you would open up a tomogram, and then you need to annotate just a tiny little bit of that data in order to set up training data and train a neural network to automate that same segmentation step for you. So in this video, I'm setting up an annotation for just a single tomogram. You can open hundreds at a time or just one. I'm setting up annotations for a microtubule and for a microtubule inner protein. So this is a purified cytoskeleton, and we see all these microtubule inner proteins inside of them. So we want a network for microtubule and for microtubule inner protein so that we can pick them, so that we can subtomogram average them. And what I'm doing here is I'm placing some boxes to say, just this square image region is what we'll use for training. And then inside of that box, I annotate the data as faithfully as I can. So here for the MIPS, that is a little bit trickier as they're more fine-grained. I think I'm forgetting one at the bottom there. And as you go along, you place a couple of boxes here and there. And after a couple of minutes of doing that, in this example, I did it for just a minute and I just did 4 boxes, which is not that many. But let's pretend the next step is to then go to the Models tab and to export these annotated boxes as a training data set. Am I doing that? Nope, sorry. Yeah, in this video. So here I say, let's create a training data set. I'm creating one for the microtubule that just exports these grayscale images that we've annotated labels for. And then I say, let's add a model. We'll call it a microtubule again. Struggling to find it. This isn't live. This is a pre-recorded video. And then we just point that model for which we can select different neural network architectures. If you're just starting out, I would recommend just using that at the default. It tends to work just fine. Select that training data we just made, click train. It takes a couple of seconds to get going. Let's see if I can skip forward. Yeah. And so at the bottom there, you can see that the network is now training. The loss is going down. And while it's training, you can scroll through the data. And this is after 10, 15 seconds of training. And you can keep an eye on what the network is learning and what it's picking up on. So very early on in training, the output is going to be a little bit horrible. All the weights are essentially initialized randomly. Sort of before you do any training, the output is just going to be noise or just all zeros. But as training goes on, you should be seeing these labels for the microtubules get a little bit better. So once you're done with that, you will find that however much effort you might have spent in the annotation step, there are always features in the data that you hadn't seen before that are causing errors in the segmentation. So the way to then go forward, and this is sort of where the concept that we wrote about in the paper of streamlining the segmentation of cryo-ET data comes into play, is you now look at the output of the model. This is after training that model a while longer, and we see that there's some output there. It kind of makes sense. This looks like the side of a microtubule, that does too. It's something that you can't really predict. You just have to have your model there, browse the data. When it makes a mistake, you copy the annotation into, or you copy the model output into the annotation tab. You improve it, and then you add some more boxes. And if you do that once and then retrain, you'll find that the network will get a lot better. You can do it again because your network will never be perfect. But if you iterate like this a couple of times, so here's another example of a wrong output that's a bit difficult to predict. It's not something I would have originally annotated, but going through it like this, you'll find that, and then you're able to improve the training data. Do it a couple of times, and you'll land on a network that is good, or at least pretty decent for your own data set. In my hands, that takes about 15 to 20, maybe 30 minutes, depending on how thorough you are. But that's with a lot of experience, of course. But you can say that in an hour, you should be able to get a pretty decent network off the ground. After that, so I retrained the network with improved training data. I then, via the command line interface, use that model to segment all my data. And this is in the rendering tab, which basically offers you a view that's very similar to what you could do in Chimera. There's even a button to go from this straight to Chimera. And so here we're looking at the output for the second or third, I think, iteration of the microtubule network and the MID the microtubule inner protein network. There are some things I'm playing around with, such as the dust level and the threshold level. But as you go through the tomogram, and you can hide the segmentation to sort of look at the actual density and then look at the segmentation output again, you see that it does a pretty decent job at finding the microtubules themselves and also the microtubule inner proteins. So once you have that in place, you can essentially, you save your model somewhere, you can decide to trust it, you can then more or less blindly apply it to your entire data set. So in this data set, we had 400 tomograms. Right now, I'm showing you 1 or 2. But we apply the networks to the whole data set, and then comes the picking step. Again, I'm showing you this in the IS GUI, but you can do all of it from the command line interface as well. So here we've got the MIP mesh rendered, and now I'm setting up a picking job. So just this data set will set a fairly random minimum particle spacing. And then after a bit, once ICE has saved a particle star file in a format compatible with warp or with Relion, and all the other data types are MRC files for input and output, so that's compatible with most software as well. You can then visualize your picks inside of your original volume. And at that point, you're basically done. So you've picked your particles via segmentation. Then comes the averaging, which is not trivial and which I'm not going to cover here, other than to say I did it with warp and rely on an M. And so segmentation-based particle picking, which you can do in a couple of different ways. Like once you have your segmentation, you can do filament tracing, which is what we did for the microtubule over here. Got to around 4 angstrom, even though it looks a little bit weird. Or blob picking, where when you have something like ribosomes or the MIPS that we were looking at, you can emplace coordinates in a slightly different way. And that's what we use for this luminal protein, which we got to 3.2 angstrom using segmentation-based picking in NICE. So now this is a bit of opinion, but I'm obviously very biased since I made the software, and so I've always been able to tweak and adjust it in such a way that it worked for me. But I really enjoy this way of working with cryo-ET data, especially when data sets get fairly large, because you essentially just play around with it. You do a little bit of annotation, you train a network, you see what it does. If you have a network that isn't great yet, but you apply it to your whole data set anyway, and then use POM, which I'll get to in a minute, to visualize all the network outputs in 2D and 3D, you will find some weird activations. And sometimes those weird activations can be actually biologically interesting. Often they are artifacts, but it's a fun way of sort of getting around your data. And doing this annotation and sort of trying to optimize a network, along the way, you stare at your tomograms for quite a while. And I'll say that staring at your tomograms is the best thing, and maybe the number one thing you need to do in cryo-ET to become familiar with what it's there and to start picking up on patterns that you hadn't seen before. So in my own experience, for instance, I played around with Clamina Mona's visual proteomics dataset for a long time. And I saw these ring-like things, which I never really realized what they were. And so I ended up ignoring them until I played around with the data for long enough. And I'd seen enough of them to realize that this was the T-ring chaperone complex. And now any other data set I look at, I spot these things instantly. So it's a cool way to sort of get used to cryo-ET data and the complexity of it. Oh, a question. I'll get to your question in a bit. So there's a question about the hardware requirements. Oh, this is the slide, right. So ICE works on Linux and on Windows, not on Mac, unfortunately, because there were some issues with CUDA and OpenGL driver, so I haven't really focused on that. Everything except for annotation, you can also do on a cluster. In fact, I always do it on the cluster. So I annotate on my Windows PC, which has a low latency, which I find a little bit smoother to use. And then when I'm done annotating, I do the training data export and the training and segmentation and the visualization on the cluster. At which point the interface is basically just there for annotation. So the question is specific hardware requirements. I've tried it on a lot of different hardware. So I also sometimes do training and the actual segmentation on my Office PC. I currently have an A1000, and then on a different PC, I don't remember what I had. The laptop that I'm presenting from, I can also run ICE there and train things there. So I'm not sure about AMD GPUs, but I think it should be fine. So I would need to look into the CUDA and TensorFlow compatibility of AMD GPUs. So I hope that answers your question. With experience, you can cook up a network pretty quickly in about 30 minutes, or let's say an hour if you haven't done it before. I plan to release some better tutorials soon with some better tutorial data. So from the microtubule dataset that I just showed you. And then originally when we published, there were only 2D networks, but you can also do 2.5D or 3D networks now. It's free and open source. We use conventional file formats. And people often ask me what the minimum particle size is, and there isn't really an answer because it just depends on what you can annotate confidently. So I personally struggle with nucleosomes, not necessarily because they're small, because I've done some other 200, 300 kilodalton things, but because I just find it difficult to comprehensively annotate every box that I place. So if you place a box somewhere, you have to annotate that unambiguously or it gets confusing for the networks. But there isn't really a minimum particle size. It depends on contrast and whatever you can annotate. Then we also have a repository online, icegravity.org, where you can download pre-trained models. And then since the original release, we have a lot of stuff added, such as model-assisted annotation, an active contouring brush, which sort of follows the brightness contrast details of whatever you're annotating. Much better CLI support, and then also synchronization with POM. So next topic is POM, which is essentially just an app, it's a browser-based app to sort of organize your gravity data and your segmentations to help you build subsets for further processing, to help you pick particles and place them back into their context, and to explore data. And it's also linked to POM. So I'll show you this video first. This is that same data set as before. And all of the videos, so these spinny videos, a bit like the camera ones, and all the static images are all prepared from the command line interface with POM. So here's a visualization of the microtubule inner proteins. The one before was the microtubules. If you have different versions of your tomograms, such as raw ones and denoised ones, you can also toggle back and forth between those. And then it shows you projection images. So if you have a lot of different segmentations for your data set, or you have a lot of different versions of your tomograms that you want to be able to compare, I find this quite useful. And then if you go into, I think I have another slide coming up. Yeah, here's another example of some slightly fancier rendering functionality that I added. So this is from a data set of HeLa cells, where we segmented the cytoplasm and mitochondria, and some of the usual bits of cellular architecture, so membranes and ribosomes and microtubules. And visualizing a dataset in this way, to me, is quite a lot more convenient than just having a fairly long list of files on a disk somewhere. And then you can go into the detail page for any tomogram, which I'll show in these videos, and have a closer look at all the segmentation values and all the 3D rendered images, but also connect the POM database to ICE. So if you are doing this iterative training of a network, what you can do is you set up your database in POM, you go to a tomogram detail page, you open a tomogram in ICE, it takes 1-2 seconds, and then you see that tomogram in ICE if you've got that running, and you can inspect the model output there. So here I'm opening a couple of different tomograms and then seeing if the MIP segmentation works well. And then I scroll through it again, and if I see something that is clearly wrong, which at this point it wasn't too bad because I'd already done a couple of iterations of this procedure. But there's something that's wrong, for instance, the slight bit of the actual tubulin over there getting annotated as a MIP. So you can erase that, improve your network, and resegment. Also, I forgot to mention, but segmenting on tomogram, it depends on the size of the field of view, but at 10 angstrom per pixel, with a field of view like this, it takes about 10 or 15 seconds to do one tomogram if you're running it on a node with 4 GPUs. And this was with RTX 3070. So that's POM. Setting up a POM database is fairly straightforward. Once you have it installed, it's just initialized to shout that you want a POM database. You then add sources. So you have to point out where your tomograms live. This is just a directory with MRC files or multiple directories with MRC files. And then you have to point out where your segmentations live, if you have them. You can also use POM to just visualize the tomograms themselves, just the density preview slices. But if you have those, you add the sources, you then type summarize. That makes POM take an inventory of all your tomograms and which segmentations you have that belong to them. And then you can render things and browse. That opens up the browser-based app that I was showing you, from where you can explore further or change your rendering settings and stuff like that. This is on github.com by nanopatterning POM. So some more details about POM. Unix, Windows, and iOS compatible, because this is really just a CLI. Actually, I say iOS compatible. May not be true, because we're using the same OpenGL drivers again. So if anyone has, also not iOS, I mean macOS. If anyone does run POM on Mac, let me know. Definitely Unix and Windows compatible. That's where I use it. Free and open source, and I maintain it actively. Then it's built to work with any segmentation source. So not just ICE or easymode, but also membrane-seg and depict and TARDIS and various other tools, as there are increasingly many. If something doesn't work straight out of the box, just post an issue on GitHub. I try to respond pretty quickly. Lately, I've been able to respond within an hour. That's just because I was in the office all day long. Usually within a couple of days, mostly within a day. Then what I've also found quite useful is data curation by building subsets. So once you're in this POM gallery, you can define a new subset, such as good tomograms, and you can go through the list and you say, I want that one, that one, that one, and that one. As if you're in a web shop, you're just clicking tomograms you like, put them in your trolley. Now they're a subset. And then when you process data in ICE or easymode or warp tools, there are arguments to point at a text file with a data subset that you want to use. So that can be useful in warp tools if you want to do reconstruction only of a couple of tomograms in a very large data set. And so the POM subsets work with ICE and easymode and warp tools. And then another thing, and I'll show you that in the context of easymode later on, is that if you have star files with all sorts of particles, and those are linked to a tomogram, which they typically are, you have your Reliant micrograph name or something like that. And then you have segmentations, and you know which particle belongs to which tomogram, belongs to which segmentation. Then you can ask questions such as, for every particle, what's the nearest membrane? Or how much mitochondrion is around the particle within a 50 nanometer radius? Or what's the distance to the void, to the lamella surface? If you have your POM database set up, asking a question like that can be fairly simple. You type POM, contextualize, you point at your star file, you then define your sampler. So in this case, I say I want to measure the distance to a membrane, the blob size for membrane. That's like the height dust function in Camera. Should at least be 1 million connected voxels, otherwise we'll consider it dust. And then we also want to measure how much of the volume surrounding my particle in a 500 angstrom radius. Oh, I'm pointing at the wrong thing, sorry. In a 500 angstrom radius. How much of that is mitochondria? You then get a value for each of your particles, and then you can stratify your particles based on that context or something like that. So I found POM especially useful when I'm dealing with really large cryo-ET data sets and many different segmentations. But you do need to have those segmentations, which you don't always do. And so I found myself retraining some networks over and over again for different data sets. So I would have a membrane network in a HeLa cell at 10 angstrom per pixel, but then I would have some other data set and it was at 13 angstrom per pixel. And I wanted to segment membranes again. And I found that between the two different data sets, the networks just didn't generalize so well. And so I thought it would be useful if we had access to more general and pre-trained networks. Basically, membrane-seg, which works really well for membranes. If we had that, but for other things that are also very common, like ribosomes or microtubules. So that's another thing, easymode, which we'll talk about now. So easymode is a collection of 26 or so general and pre-trained networks for cellular cryogravity, or for cryogravity in general. And the way that they were made is that, to get started, I collected a fairly large training data set with data from Empire and the cryogravity data portal, lots of colleagues around at the NMB, and then some people who contributed data privately. Right now, that training data set is up to about 4,500 tilt series from 30 species and about 70 different unique data contributions. And then for these 4,500 tilt series, I reconstructed them all from scratch at a common 10 angstrom per pixel and in various different flavors. So raw and denoised and isonet corrected and deeply wedge corrected. So that if you train something on that collection of 4,500 tilt series times 4 or 5 different flavors, then when you apply that network to the next data set, it should also work there. So for segmentation, we have 20 models. So ribosomes and microtubules, fairly common things. And then also some organelle masking networks like mitochondria, the nucleus, cytoplasm, nuclear envelope. And then some convenient ones like, or utility ones, sorry, like to mask ice particles and to mask the lamella boundary. Then there are also 3 general pre-trained networks for denoising, for a noise-to-noise type method, and then a deep wedge-like and an isonet 2-like method. And I'm currently working on some pre-processing models. There is already one for tilt selection online, but it doesn't always work. And then also pre-trained models for misalignment, so that you can skip that fairly expensive training step. So starting off with the denoisers, there are currently 3. The only difference between them, which I quite like, is that all the code is the same. Network architecture is entirely the same. They've just been trained on different training pairs. So if you want to denoise with any one of these 3 flavors, it's just easymode denoise. You pick your method, and then you point at your data. And the nice thing about them is that they're direct denoisers, meaning you don't have to, well, first of all, you don't have to train, which you typically do on even and odd or half 1 and half 2 half split pairs. You just apply them straight to your raw tomogram. And the way that we've done that is noise to noise is the easiest example. We basically took half split pairs for 60 different data sets, trained a single noise to noise model on that to turn the even odd pairs into denoise pairs. And then we took that same model and retrained it on raw tomograms and the corresponding denoise tomogram that we got from the previous run. And so at the end, you can apply the denoiser to a raw tomogram and get your denoise thing out. And it also works fairly well, qualitatively speaking, on novel data. I will comment, though, that statistically speaking, this is not necessarily the best way, because denoising is typically an unsupervised thing. So you train on independent half splits from your own data set. And the best way to get a denoiser that works specifically for your own data set is to just run that training loop on your own data. But with a direct denoiser, you can skip training and you can skip the half frame generation. So it saves quite a lot of computation and also memory. So see if these videos play. Up. From left to right, this is raw, noise to noise, deeply wedged, and isonet 2. The deeply wedged and isonet 2 models are distilled from 50 separate deeply wedged and isonet 2 training runs on individual data sets, and then pooling all that data and training one larger unit on the pre-denoise training pairs, if that makes sense. If not, let me know. Those are online, so you could use them. Actually, when you use them, so this is basically just throwing compute at a problem, and pre-training a deeply wedged or an isonet 2 thing is convenient. If you do use them, remember that behind the scenes, you're really using deeply wedged and isonet 2. And then the main point of easymode is to render the biological stuff that is in tomograms, and tomograms are often very complex and there's a lot going on, to render that computationally accessible, so that you can ask questions like, I have this set of particles, which ones are in a mitochondrion and which ones aren't? here are two examples of input and output, both from datasets that I got from Empire. So this is from Herman Fung, and this is from Patrick Hoffman. At the top, it's a HeLa cell that was not trained on, and then using pre-trained networks for these various features at the bottom, you can turn this into that. Or below is a dictyostelium discoideum cell, same networks, same everything. You type easymode segment, long list of features that you want to segment, it finds the weights that it needs, and then it gives you these outputs. It doesn't give you the images, it gives you segmented MRC files. So what you can do with those, having computational access to the data, to the biological features, is first of all, filtering particles for a specific compartment. So in this example, we took this data set of HeLa cells, which were treated with a compound I did forget the name of, but that induces folding stress inside of mitochondria by Kenneth Aces et al. And we used Python at first to pick mitochondrial heat shock proteins, which gave us a set of about 20,000 particles or so. And then we used easymode segmentation of mitochondria, ribosomes, membranes, and these mitochondrial granules to refine the particle set. So we said if a particle isn't inside of a mitochondrion, or it is too close to a membrane, or it's too close to a ribosome or a granule, we discard it. And that left us with only 3,500 particles from starting 22,000. But what you find is that using that biological prior information, which sometimes it can be difficult to express in computationally accessible terms, you of course get a much better map because you now have particles that actually make sense for the thing that you're trying to average. Another example is classifying or is measuring the distance from any particle to the edge of the lamella. So it's been shown by a couple of papers, including by Mart and Teitel from the MPI Frankfurt, that particles closer to the edges of the lamella have offered less high frequency information for subtopic gravities due to ion implantation and that sort of stuff. So if you can simply measure using a pre-trained network to annotate the boundary of the sample and then measure the distance from every particle to that boundary, and then stratify your particles, and say, so in this case, the median distance to the lamella surface, measured for every ribosome that we found, was 43 nanometers. So we split the particle set in half. The ones that are far from the surface, which should offer a better resolution and go to 4.2 angstrom, and then the ones closer to the surface, which should be damaged, or that would be damaged, and offered lower resolution. And then another thing that I think is cool, this is with a dataset from Force and Gao from David Barford's group, also at my institute. He purified mitotic spindles, and we know biologically that the microtubules in mitotic spindles originate at a gamma tubulin ring complex, which is found at the spindle pole body. So if you want to find gamma Turk particles inside of a cell, what you would do if you could sort of pseudocode it is you go into the sample, you find a microtubule, you measure the polarity of that microtubule, you now walk towards the minus end, you're in the lamella, so you have to check, does the lamella stop here? Because maybe the minus end has actually been destroyed. If not, if still in the lamella, walk to the minus end and place a coordinate there, because that should be a gamma tubulin ring complex. So that's what we did here, using easymode to segment the microtubules and then to trace them as a spline at the sample coordinates along them, then averaging every individual microtubule, walking towards the minus end. And if that minus end was still within the slab, then we put the coordinate there, and then we averaged all these coordinates, which, based on just the geometry and the biological prior information, we knew should be at the minus end. It still took a bit of classification, and in the end, about 1/3 of the particles averaged to this gamma tubulin ring complex class. So this bit over here is just a regular bit of 13 protofilament microtubule, and this is that gamma tubulin ring complex. Then an example from my own work. So for my postdoc, I work on, well, one half software development, and then the other half, this metabolic system here. So this is purine synthesis in mammals. Is a cascade organized by about 8 different proteins. IMPDH, inosine monophosphate dehydrogenase, is the rate-limiting enzyme in that pathway, and it's present in fairly high concentrations compared to the other ones. And when you inhibit it with a drug called mycophenolic acid, it forms octamers. The octamers form filaments, and the filaments come together to form bundles. And that's what you can see happen in this video, where we have IMPDH with a fusion of a GFP treated with mycophenolic acid. And the reason we study this is because we believe that these filament bundles aren't enriched just in IMPDH, but that there are also other enzymes that are involved in purine synthesis present in those bundles. Now, based on the literature, there are reasons to believe that a thing called metabolic channeling occurs in purine synthesis, where enzyme A hands off the metabolite directly to enzyme B through some structured pathway or some way of organizing spatiotemporal proximity, rather than the one being over there in the cytoplasm, the other one being there, and relying on luck. So some of these reasons are that the intermediates are toxic or highly unstable. And when we co-stain for different enzymes in the same pathway, so PRPS, GMPS, and ATIC, so that one, and that one, we find that they're also enriched in these filament bundles, and then CTPS, CTP synthase, is also in there. So in this case, we had already found IMPDH in, well, we spotted it in one data set of the easymode training data collection, and then annotated it there, and then ran that network against the whole training data set. And by the end, we found IMPDH filaments, which you can see in this tomogram over here. It's these sort of discs on a chain. It looks a little bit like a spinal cord. We found them in 9 different data sets that are in the easymode training data collection, in mice and in human cells exclusively. So what we did is we then used that network and used it on our own data set of, by then, about 1,000 tomograms collected in HeLa cells. And we found about 20 of these tomograms that actually contained these IMPDH filaments or even bundles. So here's a visualization of easymode network outputs that we used to then. So after finding IMPDH, we made a subset of tomograms of interest and threw some more networks at those. Here's another one from one of the easymode training data sets, where you see more of that IMPDH coming into view right now. And then here, yeah, this is one of the ones where you see an actual bundle of IMPDH. And so in a single tomogram, you get 15,000 or so particles, with all of which are octamers. So you get over 100,000 particles in a single tomogram. So that's what I use easymode for myself, and POM as well. Here is an average of that filament as an enzyme as a filament. This is the map we currently have. And so we're looking into basically improving our tomogram quality using maybe misalignment and some more eyes on it too. And then what we want to try is to see if we can find any of these other enzymes that we believe are there. We can find them inside the bundles as well. So some details about easymode. It's built to run no longer on my Windows laptop, but on an HPC cluster. Again, it's free and open source, and I maintain it. And I intend to polish up the networks over time, and also to add networks for additional features. One way in which you can help is if you have easymode installed, and if you run the segmentation for any sort of feature and find that the output isn't great, you can type easymode report -- tomogram and then point at the tomogram where the failure occurred. You can add a comment to say microtubules didn't segment well, something like that. It gets uploaded to a private server, and then I can ingest that for the next training run, and hopefully over time make the networks a bit better. So the models are general because they've been trained on a fairly large and also diverse training data set. So this includes in vitro reconstituted stuff, purified things, mostly gallium fib male lamella, some symovis, some whole cell periphery, plasma fib milled stuff. And a lot of that data came from Empire and the Priority Data Portal by the CZII, without which none of this would have been possible. So on processing time, large things like the mitochondria or the cytoplasm, those segment at 50 angstrom per pixel. So that's pretty fast. It takes about 1 to 10 seconds per tomogram. This measured on a 4 GPU node. And then the finer grained things like ribosomes and microtubules, which we segment at 10 angstrom per pixel, they take a little bit longer, between 15 seconds. So for the tomograms I showed before, tomograms of about that field of view size, it's about 15 seconds. For the larger tomograms, if you acquire at 2.5 to maybe 3 angstrom per pixel, and you have a much bigger field of view size, then it can take a bit longer. They were validated by subtomogram averaging on unseen data. And then the reasoning is if we get to a high resolution microtubule or ribosome or T-ring chaperone complex on data that's never been seen before, then the picks are good enough for averaging, then the output is good enough for us, and or precision and recall measurements. So for something like mitochondrion or the nuclear envelope, we can't average, so we do it with more conventional metrics. And then finally, it's not a tool for picking arbitrary targets. So if you want to look at ARP2, 3, if I remember the name correctly, this cross-linker for actin, that is more niche than microtubules, and so there probably won't be a pre-trained network for that particular complex. But in some cases, you can still use easymode. So if you're specifically looking at actin cross-linkers, for instance, if you are able to segment the actin and then trace the filaments and then find cross-sections between filaments, something like that, it might still help. But overall, it's not a tool for picking arbitrary targets. And then we have documentation online at ngfast.github.io/easymode. And we also have 7 tutorials there for the different subtomogram averaging tests that we did for the paper. So the ones I showed about the heat-shock protein and the lamella to ribosome distance, the gamma-tubulin ring complex, they're online if you want to try something like that. Yeah, and so another note about the easymode training data collection. This is the people who supported compiling that data collection. So EM facility and LMB scientific computing. And then these are the people who contributed data. So a fair number of individuals. And then I think we had about 30 data sets from Empire and about 10 from the CryoT data portal. This slide is just here to say uploading your data to a server like this is super useful. If you have any data left over, even if it's horrible, please consider uploading it, because even a completely misaligned, completely artifactual tomogram is still super useful for training as a counterexample to what genuine feeders look like, for instance. This video here, does that play? No, represents Empire and me. And this cat is the real POM and inspiration for the logos. So all that said, ICE, if you want to train your own network, easymode if you don't want to train your own network. But if you are looking at something we don't have a pre-trained network for, I suggest starting with ICE. And then POM is this tool for visualization and curation and contextualization. And I did that work over the last 3 years, originally with Tom Sharp and Leonard Fortman at Leiden University Medical Center as my PhD advisors, and Leonie Abenstein, who is now in Vienna. And then POM was with Tom and Leonard again, and Matteo Allegretti, who I work with currently. And then easymode was with Tom Hill, Alistair Burt, and Matteo Allegretti. Oh, and that was my last slide. So that's it. I'll take any questions.
Oh, thank you very much. So we have 2 questions in Q&A already, and we have people who found the applause button. I haven't found it yet because I'm used to Zoom. So yeah, if folks have more questions, please feel free, use the Q&A button, send them in chat or raise your hand, and we can ask them yourselves.
Right. Okay. I see the 2 questions now. They're a bit long, so I'll have to read them in silence. Oh, Spencer, do you have a question? Or you have your hand raised?
Can you hear me now?
Yep.
Good. Raising your hand gets you microphone privileges.
I flipped the switch.
Oh, great. Thank you, Pete. Two things came to mind. First of all is about POM contextualize. Can you give a more context for what commands are available for POM contextualize? You mentioned, I think, just measuring radius. Can you do things with angles? What commands are available? What
Hmm.
documentation is there for that?
I think if you want to look a little bit more in depth at correlations between particle A's orientation and then particle B's orientation, etc., I would get started with Beate Turanova's Tango, probably. I haven't yet used it myself. I've seen her present it twice, and it looks super useful. Because that is currently not in POM. And also their framework is to represent any sort of feature as a point or an oriented point. Whereas in POM, your particles are represented as an oriented point. Your segmentations are just meshes or MRC volumes essentially. And so the measurements that you can do in POM is you can measure the distance from a particle to a mesh. You can measure the distance from a particle offset along its primary principal axis to that mesh, or a local sample of the environment and then the average segmentation values in that environment. I'm not sure if an oriented point type of description is always super useful. For instance, something like a mitochondrion would turn into a fair number of points if you sample them very densely. Maybe that's not a problem. But yeah, does that answer your question?
Yeah, it does.
Perhaps not.
Thank you. And I've used
Oh,
the oriented point clouds in CryoCat from the Turnova lab,
oh yeah.
and they're very nice, work very well. And so that mentally was comparing POM contextualized to that. So I'm glad that's what made you
Yeah,
think of it too.
yeah. There's a bit of overlap there, I think. And then POM is a bit simpler
Right. It
at the
seems, yeah,
moment.
CryoCat is sort of the heavy lifter if you need to do some real rigorous, exactly what angle and, ray tracing
Yeah,
at certain subsets of degrees, whereas POM is a little more, like you said, built for a large data set for doing summary stuff.
yeah. Okay,
Can I
then.
ask,
Oh,
the last thing was you mentioned sort of statistically, in terms of statistical rigor, denoiser is probably not as statistically rigorous since you're not using a model trained on your particular data.
mhm.
How does the output compare if you take ISONET 2 and train it on a split tilt series versus
Well, yeah,
using,
that's why I say that qualitatively to me, it basically looks the same.
okay,
I just don't want to claim that it's better because it technically shouldn't be. But I no longer I still use ISONET all the time through the lens of this pre-trained thing, but all the credit to ISONET 2. But I find it much more convenient to just skip the training and uh, also to not generate the half maps because especially with this training data collection, but I think everyone, once you start doing things at a slightly larger scale, you're just running into storage problems all the time, which can be annoying, but it's also a realistic problem. So right now I'm at 40 terabytes of stuff. Getting more is just very expensive. And so avoiding the generation of the half maps, even if you can discard them after, is quite convenient. Yeah, could probably quantify that a little bit better. But the main reason I made the pre-trained denoisers is, I guess, also to save memory. It's so that during the training of the segmentation models in easymode, all I need to have is the raw subtomogram, and then during training, as an augmentation, we can pull that subtomogram through a noise to noise or deep denoiser or ISONET 2 denoiser to get the other flavor out. Does that make sense? Maybe.
you're
And
running
that's in
denoising
hope of.
on subtomograms, not full tomograms, you're saying?
Yeah, well, for the things I showed in the presentation, those are full tomograms. But
Right.
for when I'm training the ribosome segmentation network, for instance, I have the raw subtomograms on a disk. They're always a little bit larger than the packs that I actually train on. And then for augmentation, in order to land on a more general network, I then turn that
Yeah.
raw one into a denoised or a ISONET 2 corrected one. And I found that a general pre-trained network there was very convenient because I was dealing with a lot of different data sets and couldn't store all the different denoised variants all the time. So that's why I originally trained up these networks, and then I found that yeah, so this is opinion, and I should maybe quantify that better, but that the performance to me is good, good enough that I haven't really looked back, but sometimes maybe I should.
Yeah, great. Thank you.
Okay, we have two questions in the Q&A. The first is, you
So I
described
think
what appears
the
to be...
Q&A questions, I think we have moved to voice chat. And so I will turn it over to D'Souza1 for unmute yourself and ask your questions if you would.
Hey, this is Duncan. Can you hear me?
Yep.
Okay, great. Yeah, so sorry for the lengthiness, but a little bit complex. So the first one is, as we all looked at our data sets and like learned over time how to select our particles and what they look like and that sort of thing, is there anything that you can think of that will help train our AIs aside from large context? I mean, we have a giant context of what things should look like and kind of what we're looking at in sizes and stuff like that we can't just plug into the model easily. But are there other things you've thought of that might be useful in getting your model to behave better just from the way you learn? Because they seem quite parallel in a way.
So the parallel you're describing, is that the one between networks learning to detect stuff and me staring at data and then detecting
That's
stuff
right.
eventually? Yeah.
Yep.
Okay. Good question. So ISONET 2, I'm very impressed by ISONET 2 when it comes to certain things. So for just improving the contrast, it's brilliant. For well, let's keep it there. Improving the contrast, it's great. And so that came out as I was halfway or 2/3 of the way through compiling the training data and training up the networks. So originally I didn't include it as an augmentation. And some users found that for nuclear pore complexes or microtubules, if you apply the network to your raw tomograms or to your noise-to-noise denoise ones, the output was better than applying it to an ISONET 2 denoise one. Fair enough. Sort of the flavor of the tomogram is fairly different, but I think it goes to show that the way the data looks is changing fairly rapidly. I still think it's not going to change that much. A raw tomogram is just a raw tomogram, and that'll probably stay the same until we get massive improvements in detectors or until laser faceplates hit the street, so to say. But these sorts of augmentations that enhance the contrast, I think if I'd done all the original annotation, so a fair amount of manual annotation went into generating the pseudo labels for the later on training in easymode. If I had done that on ISONET 2 denoise things, that would have definitely been easier. Yeah.
Okay.
But other ways to improve the interpretability of the data, I'm not sure at the moment.
Yeah, and we're not there yet in applying the current ChatGPT style contextual AI models to saying things like choose all the proteins in this density. There's no way to learn that as easily as
in that way. Sorry, I was just thinking about the data going in. As far as the machine learning goes, I mean, easymode is super simple. It's basically just the same neural network as used in membrane seg, because that seems to work well for membranes.
Yeah.
And then it uses very sparsely 2D or 2.5D annotated things in ICE. So I have a POM database somewhere with the 4,500 tomograms. I go back and forth between that and ICE to do a little bit of annotation, and then I bootstrap all the 3D training labels from there. But it's as it was described in I think the CSRI machine learning competition paper, it was called vintage machine learning, 2.5D and 3D units. And that's true. And I've seen some interesting work on using large pre-trained transformers to sort of embed the context of a tomogram and then to give that as a thing. Cool.
Do you have a link to that paper?
I'm afraid it's not online yet. But
okay. All right. Well, I'll look forward to that.
Yeah. Yeah. Well, there was an earlier paper, so I forgot the details about that one. Cryo VIT, I think, so vision transformers for tomogram segmentation, where they looked at mitochondria. I think if you Google that, you'll find something. So on the machine learning side, I'm sure we can do a lot better. I hope we can do a lot better than this. But
Well,
I
if
was
you've got
mainly
a,
interested in.
I was going to say, if you've got about $1 or $2 billion, then I can build a giant data center for all of our EM data, and we'll do what the ChatGPT did, which is just increase the amount of data that it's looking at
Yeah,
across all
sure. But
domains,
now,
and it'll get a
but
lot
now
better.
you're running into now you've made it a social problem because where are you getting the data from? I would happily send you everything that I have here, but I can't because some of it was contributed privately, with the understanding that I would not reshare it, which I will honor. I mean, I would love it if basically if Thermo Fisher, or if let's say Thermo Fisher decided that between your Falcon and your server, you now have a
Yeah,
box that uploads everything to your $2 billion data center.
yeah,
I would be game if we all did it.
yep.
But yeah,
I'd be fine with something like that, or you publish and you just give all the data, including all the junk, which I think is tossed and is unfortunate because that is excellent for negatives.
yeah.
And that's a lot of the learning. But my last question here, I don't want to take too much time, is do we know what we might be missing by doing these denoising operations and whatnot via AI? There's the typographical examples of you've got this lovely picture and whatnot, and you white out some of the areas and you have the AI fix it, right? Does an excellent job of filling stuff in, except when there's a missing like vase on a table, it just doesn't know it's there. It's impossible for it to know,
Mm-hmm.
So
Yeah.
of course it doesn't have it there. We have a missing wedge, and that's not there, but there is a certain amount of context of like protein has a certain level of structure. The missing wedge does a blurring in a certain direction. You can back that out from there's other modes of information that are sort of contained in there that might fill that sort of thing out. One could imagine, right? Even though the data is actually missing in that direction for that tomogram.
Yeah.
But other things you can imagine that we might keep in the back of our heads that would be missing, like the vase, that the AI will never be able to fix. Things we're interested in,
Yeah.
We're not interested
Other
in the shop
other
notes.
than the 20,000 sub 200 kilodalton complexes that we all ignore, maybe.
Oh, right, yeah, of course.
I'm not sure.
Mm-hmm.
I'm not sure I have any insight there. One comment on the missing wedge, correct,
Yeah.
or the missing wedge in painting, essentially, is what it is. I would guess that maybe there you're a little bit more at risk of when you're using a pre-trained thing, and
Yeah.
now you have a data set that's a little more unique. Now it's going to look, it will have structural priors that your own data would not have offered, and so it would maybe paint in the wedge in a way that would not have happened if you had retrained something from scratch in your own data.
Oh, that's a good point. Yeah, so if it's a different contextual thing in a cell, and maybe it's not a cell, maybe it's a material or something, it's going to fill it in, perhaps with bad priors. Okay.
Yeah. But still, for contrast enhancement and just yourself looking at the data, it's still super useful.
Oh, it does. It's such an amazing job. Yeah.
Yeah, but it's a good question, but I'm afraid I lack any insight.
Okay. I appreciate it. I figure you guys are at the very cutting edge, so if there is insight, you would have it.
I'm not sure about that, but maybe if there is insight to be had, I'm sure it'll be published soon.
Excellent. Well, thank you so much.
Yep.
I guess another call for questions. Folks can raise your hands, use the Q&A. I have a question about which this may be an ACE question, this may be a vintage machine learning question, or it may be a naive question, but I will ask it anyway. When you were talking about using ICE to, manually annotate your data, do some training, manually annotate your data, I was reminded about some of the very old machine learning algorithms where they called it active learning, where the algorithm was basically designed to do that, and it would flag regions of the data for the regions where it was most confused about and say, please take a look at this because this will help discriminate my classifier the best. Is this something that's built into ACE? Is this something that has been renamed into a newer tool in machine learning that I'm not sure what they call it nowadays? Or
No, that is not So the automated digging up of image regions that the model was the worst at segmenting, that's not in there currently. I just do it via visualization and looking at many different 3D renders at the same time and sort of seeing what's up. Could be useful. It also could be relatively straightforward. One way of doing it is just segment in various different augmented ways, and then wherever the consensus is weakest, you would flag that. Might actually consider doing that. But no, it's not in there. I also wouldn't know if they've rebranded this as something modern and fancy since when the unit was invented.
Yeah, I mean, it sounds kind of like the the reinforcement with human feedback thing. It's like that sounds kind of like that, but like this. I thought this had a name before, but I'm also not a machine learning expert, so
I'm just saying that that specific, like the automation of finding the image regions which you would best spend extra time annotating, that hasn't been annotated. But the going back and forth between checking the model output and then seeing what you have to improve. Sure, that has a name. I called it model assisted annotation. It must have a better name.
No, it's probably got multiple names, but I'll defer to yours because you wrote it.
Cool, thanks. I just want to take this opportunity to say it sounds like a lot of work when I said that you do 3 iterations of going back and forth between the data, etc. But going back and forth between checking the output and then going back to the annotation, you just press 1 or 2 to toggle between the different tabs. So it really is, once you get the hang of it, and that's why I probably need to make some better tutorials. There are YouTube videos by now a little bit old. These are from when I was quite a bit worse at using the software myself. So I've learned by just doing. So I want to make some better tutorials to maybe help people get into that mode of doing the iterative refinement, because it can really be pretty easy eventually. And I find it nice. But again, I'm super biased.
Well, once you pick up the knack of something, you always get faster at it.
Yep.
All right. I guess will do one last call for questions. Going once, going twice. Looks like no more. I'm sure I could ramble on with more questions, but we've hit the top of the hour.
One real quick question.
Okay.
What for your OS that you're running on your laptop and whatnot? Is it Windows or Mac? Do you just run straight native Linux? What do you tend to do to get all this stuff working?
No. I wish. So when I started at the EMBO, everyone's using Linux here, and I did too. And I wish I could have stuck to it, but Microsoft is just—they've got me. I couldn't get used to OpenOffice. I had to use Microsoft Word, so here I am on a Windows laptop. So yeah, Windows for annotation, but you can do that on Linux just fine. That's what I did for the first 6 months before and then most of the actual processing I do on a cluster, which is all Unix, Linux, something. Yeah.
All right, same thing I do. All right, thanks.
Sure. All right, that does it then, I suppose.
All right. Well, thank you again for joining us, and apologies for getting your last name wrong. And
no, sorry. When I said, I instantly regretted it when I called you out on it. Sorry.
No, I got it wrong. So.
Okay, well, that's fine. Okay, well, thanks for showing up, everyone, and listening. And if anyone has any questions, please don't email me, but just post something on GitHub so that other people get to reuse the answer. Or do email me if you want to, but I prefer GitHub. Yeah. All right. Bye-bye.
All right. Thanks, everybody.